All right. Before I get started, I just want to say thank you to David and all the people who organized. It's been a really, really cool, fun conference. Also, I want to say big thanks to my co-authors and collaborators listed here. They did a ton of work building a foundation to allow me to present on what I'm going to present, and the funding sources listed here. My name's Dylan. I'm a postdoc in Heidi Sossack's group, and I'm going to talk about work we're doing trying to develop high-throughput approaches to quantify diatom parasitism. So if you're like me and not a parasitologist, I thought maybe we could start with some trait-based definitions of what defines a parasite. So according to some prominent parasitologists, the trait that distinguishes a parasite from a more traditional predator is that a parasite feeds on a single individual during a given phase of its life cycle, whereas a predator feeds on multiple individuals at all stages of its life cycle. Another trait that tends to separate parasites from predators, for the most part, is that parasites tend to be smaller than the organisms they consume, whereas predators tend to be larger. And if we think about what this means for planktonic ecosystems, where we typically assume a size-structured food web, if parasitism is an important process in these systems, this kind of reshape how we think about the flows of organic matter through marine ecosystems. And there is a growing body of evidence that suggests that parasitism may be an important process that is often overlooked in planktonic food webs. So these figures come from the Terra Oceans Project. So they basically did a global scale DNA sequencing survey. And one of the really striking findings from this survey was the extremely high abundance and diversity of what we think are eukaryotic parasites. We don't know exactly if they're parasites, but they're at least genetically related to known parasites. And in this figure on the left, you can see that these parasites tend to significantly correlate with a diverse collection of both zooplankton and phytoplankton, which suggests that they may be interacting and parasitizing a diverse group of other plankton. And if you've spent any time looking at plankton images like we do in the Sossack lab, you've probably seen a pretty diverse array of diatoms interacting with what appear to be small eukaryotic parasites. So you can see these, let's see if I can use this, these small flagellates coming up and attaching to the outside of a diatom frustule. And you can also see there's a decent number of these other diatoms with very, very strange intracellular morphologies that may suggest they're being parasitized. So clearly, we should probably think about how we can quantify both diatom and phytoplankton parasitism across large spatiotemporal scales, so we can begin to understand how prevalent these interactions are, as well as how they're governed by oceanographic forcings. And so one way we might do that is through imaging flow cytobot, or IFCB, which Heidi just beautifully introduced. So I'm just going to say IFCB generates a lot of images, and some of them look like those shown on the left here, where you can see this. And so these images come from kind of a seminal study at MVCO, which Heidi also introduced. MVCO shown with the star on the map there. And so in this study, which was done by folks in Heidi's group and led by Emily Peacock, they identified this diatom you see in this images based on its morphology as Guinardia delicatula, which is a biomass-dominant diatom at MVCO. And by lining up this infection cycle, you can see across these various images here with previous microscopy studies, they were able to identify this parasite as a cercozoan called Cryothecomonas aestivalis. And that was later confirmed with DNA sequencing on an isolated host parasite system. So this is a nice model system we can use to begin to study infection dynamics. And so also in this kind of seminal study, they manually annotated a whole bunch of images to construct a nice time series here, both of our host diatom's abundance and its infection dynamics. So as we know, there's a lot more image data out there, now over 1 billion images at MVCO. There's another 10 million or so across a broader spatial domain here on the Northeast Shelf, available through various cruises. And this is just from Heidi's group. There's other people using these tools. So we clearly need to develop automated approaches. And one way Heidi's group has been doing that, and now I'm trying to do, is through the use of these convolutional neural networks, or CNNs. And so the current iteration of CNN that we're using can separate about 150 different classes of images, which includes both healthy and infected G. delicatula. And an important thing to know about these CNNs that's going to be relevant to some of the results I'm going to show is that to make your image classifications, for each image, they spit out a series of scores that are essentially probabilities that each image belongs to each class. So that's kind of what this cartoon is showing. So that gives us some flexibility in how we implement these CNNs to best classify different groups of organisms. And we have this beautiful manually annotated data set that has more than 70,000 images from about 1,600 samples that we can use to optimize and validate our classifiers. So let's see how it works. So here I'm showing the results of the CNN on the vertical axis and against manual counts on the horizontal axis. This is for healthy, so not infected G. delicatula. These two panels show the same thing, linear and log scale, so you can see that big dynamic range there. Clearly, it works extremely well and quantifying non-infected host cells. There's some bias here that we may circle back and try to correct later, but this is certainly good enough to do oceanography with, and what we're going to move forward with today. And so if we look at how the CNN performs on quantifying infected host cells, we see some pretty substantial bias in the CNN. Again, we have the automated counts on the vertical axis against the manual on the horizontal. So the CNN tends to overestimate the concentration of infected cells. So we can play around with our CNN scores a little bit to try to improve this. We've done that by implementing some score thresholds, which improve things a little bit, but still a lot of bias in there. But if we use a classification approach that accounts for the distribution of scores across all classes, we end up with much improved fit. You can see in the blue here. And again, this certainly looks good enough to start doing oceanography and ecology with. Okay, so now we have an optimized classifier for both healthy and infected host diatoms. And we can see how we do in predicting infection rates. So here we have our points colored according to the total concentration of our host diatom, where blue indicates low concentrations of the host, purple indicates high. Although the CNN does not perform particularly well at low host abundance, when the host is reasonably abundant, greater than about 10 per mil, we do a very good job quantifying infection rates. And since you may not be familiar with phytoplankton parasitism, I thought it'd be important to point out that our infection rates go up to about 50% when the diatom is that abundant. So this could represent a significant loss term, at least for this particular diatom population. So now we can apply our classifier to all that image data I mentioned and create what I think is a pretty beautiful time series of parasitism in this model system. So here on top, we're looking at the total concentration of our host diatom. The points are again colored according to the infection rates. And on the bottom, we're looking at temperature, and my gray shading regions didn't come out, I guess. But basically, where you see these regions in the winter here, that's indicating periods where the water temperature was colder than 4 degrees Celsius. And so this has been previously observed to provide sort of a refuge from parasitism, where parasitism in this system is suppressed. And so we see within those shaded regions, all our points are blue, indicating low infection rates. And we also see our host diatom tends to bloom during these periods or immediately following these periods, which suggests that this release from parasitism may be an important factor governing when this diatom blooms. So that was pretty exciting. And we can look a little deeper here and try to come up with new hypotheses to explain these infection dynamics. So here we're looking at the annual cycle in our host diatom, again, colored according to infection rate alongside the annual cycle and temperature. And one interesting feature that pops out here is in addition to our wintertime bloom, infection seems to remain very low in the spring and into the summer, even after temperatures ramp up and would, in theory, allow infection. So this raises a bunch of questions as to why we see this pattern. Obviously, this is kind of very early days, so I don't have a clear answer yet, but one possible explanation is an interaction with light availability. So on the left now, we have irradiance on the horizontal axis versus temperature on the vertical. Our points are colored according to host abundance again, and with those black circles indicate periods where we have both high host abundance and high infection. And so we can see we only really tend to see these large infection events when temperature is high and irradiance is relatively low. For some reason, we don't see any infection going on or any substantial infection when irradiance is high. And so there could be other environmental correlates or biological processes underpinning this, but certainly an interesting hypothesis we could begin to test by collaborating with modelers or experimentalists, say. Okay, so that's pretty cool. So obviously a lot more work to do to explore this model system, but we've also been thinking about how we can scale these approaches to the broader diatom community preferably without looking through another couple million images. So one way we're trying to do that is through the use of these genetic covariation networks, with the idea being maybe we can use the genetics to guide manual analyses to search for additional interacting hosts and parasites. So you're looking at a covariation network here on the left, where each of those green squares is a genetically distinct diatom species, and each of the circles is a genetically distinct cercozoan species. And so cercozoans are the lineage that our host, our model parasite comes from. So it seemed like a logical place to start. You can see we recover, and I should have said these black lines indicate significant covariation between two species. So you can see we recover a significant correlation between our model system, our Guinardia host and Cryothecomonas parasite. So that suggests this may provide a feasible path to do what we want to do. So we can try to look for other examples here. So we'll try to look for some interactions between this Didelum brightwelli diatom and a genetically distinct species of Cryothecomonas. So we'll use our our DNA sequence abundance profiles of both hosts and parasites, which are shown with the dots here, alongside our IFCB classifiers estimates of the host abundance to find events where we can zoom in and look for signs of parasitism. You can see, if we zoom in on this event, we see some very interesting images here that may indicate parasitism, although I recently learned that didylium also kind of looks like this when it's undergoing gametogenesis and getting ready to sexually reproduce. So that's a new obstacle that we're kind of trying to figure out how to work through. And another cool thing about this idea is that we can expand to a much broader diversity of potential parasites. So here we're looking at two Cattosaurus species interacting with the very diverse collection of three different potential parasites. So same story where we can use the abundance profiles to find events to look into and find some interesting evidence that may suggest that Cattosaurus is being parasitized by perhaps a zoosporic fungi, which has actually been observed before. So at least some exciting potential. Again, it's very much under development. So I'll conclude there and just summarize. So we're pretty excited about these automated image classifiers, applying them to look more into the dynamics of infection in this model system. Again, this is very much ongoing, so I'm evaluating a couple different CNN configurations and trying to get the best estimates of infection we can get. And then there's a lot of fun ecology and oceanography to do once we do that. As far as scaling out to the broader diatom community, we probably need to resolve a complete infection cycle that resembles something that's been observed in the literature before, as we've done, or as Emily's done with our model system. But yeah, I'd love to hear your thoughts on that as well as trade ecologists. So I'll end there and say thank you, and please feel free to contact me if you'd like to talk more after the conference.
So when David put up all the exciting things he was expecting to hear from this thing, parasites, I mean, how often have we heard about parasites? Wonderful, wonderful talk. Thank you. Questions? Okay. Kyle, do you want to unmute?
Sure. Thanks. That was super interesting. One thing I was wondering, you showed that Terra Oceans data at the start, and one of the strange or intriguing things about that data set is that the putative parasitic organisms in those smaller size fractions appear to be more abundant than the free living non-parasitic phytoplankton and other eukaryotes. And it's kind of unclear how much of that is true versus bias in the methods. So I was just wondering, from your data, do you have any sense of how those numbers might line up?
Just in terms of comparing the abundance of parasites to hosts?
Well, I'm thinking more, since you're looking at what seems to be a fairly abundant host parasite system, how are the numbers of these small parasites compared to the numbers of similarly sized protists in the system that are probably free living non-parasites?
That's a great question. I don't know. If I certainly haven't looked into that really, and we can't. There's a big parts of the parasite's life cycle that we're missing in these images, where we're focusing on the microplankton that are being infected. So it's kind of hard to say from what I've been doing, but I've certainly in the amplicon data, these putative parasites appear to dominate oftentimes the smaller size fraction as far as I've seen, but that's really all I can offer on that.
I guess I'll steal the question. A sort of related question. I mean, because you're seeing these optically, if when you count the parasite, do you count it because it's free, or do you also count them when they're inside their hosts? So when you're showing the abundances, is it abundances of free living parasites, or is it the abundances that are within hosts, or both?
It actually we're not quantifying parasites at all. We're just quantifying infected hosts.
Okay, so you're not seeing the parasites singly. You're just seeing them within the hosts. Okay,
exactly.
thanks.
Yeah.
Sorry, Joe.
Very cool talk. Being eaten from inside doesn't sound like fun, but just a real kind of technical question, I guess, in terms of the image processing. When you go through and before you know there's a parasite that is infecting a particular host, do you have to go back and detrain some of the algorithms to say, oh, this was not just this particular algae, but it was actually algae with parasite? Or is there any issues like that kind of pop up?
You can. So Heidi's group has this awesome infrastructure where they annotate images as like a particular class of diatom, say, and then they can tag the images with, interacting with external detritus or parasites or what have you. So, and then when we're training the classifiers, you can kind of pick and choose what classes you include that the classifier learns to distinguish, and then it will distinguish kind of healthy versus infected, or you can say, no, don't distinguish infected, just call them the host diatom, and then we'll distinguish them later. And so that's kind of stuff we're kind of evaluating how changing some of those parameters might improve these outcomes that I showed. But
I guess I was kind of wondering, before people know that there is, that I guess you could just flag it as being odd.
Right. Yeah. So that's how they have that infrastructure built, which some of my co-authors did, that allows you to kind of flag that for later,
cool.
Which is really nice. Yeah.
Yeah, that was fascinating. I was super interested in your sort of, I don't want to say speculation, but the hint that there might be less parasitism at high irradiance and low temperature. And I understand that you may be hesitant to sort of speculate, but do you have any thoughts on why, like physiologically, that might be that parasitism is lower under those conditions?
I don't. I would kind of almost suspect that the irradiance is almost just a correlate of the seasonal cycle, and it might have to do more with the parasites biology. There's also been studies in different host parasite systems that have shown an impact of irradiance on parasitism. So I thought it'd be worth looking at, and sure enough, there's a pattern there. So it's hard, when you're doing this kind of observational stuff, it's hard to really pin down, especially when we know so little about the parasites biology. But
is there a culture system for this? Is there a lab model system?
Heidi's group, I think, has isolated it before, but I don't think I think it dies after.
It's very tricky to keep the whole diatom if they don't have sexual culture.
Yeah, sorry. We've been able to isolate them repeatedly, but it's tricky because we can't keep the host a long time because they don't have sexual reproduction in our cultures. So we end up losing the host and have to re-isolate them. But we were able to do it several times. One interesting anecdote is the first time we isolated it, there was a big bloom and it was cold water. We got the host. Everything was going great. The host was growing really well. We warmed the culture up to get it to grow faster, and the parasite came out of the woodwork and killed the entire culture. So they were somehow hanging out in there waiting for it to get warm. It's interesting. There's a lot of parasite systems, I think, where low temperature inhibits the parasitic activity. So it's maybe not surprising.
All right.
So I was wondering, with using the genetic correlates, whether you always expect to later be able to pick those things out of the images, or whether in some cases with using the genetic data, you're finding potential parasitic interactions that you wouldn't actually be able to see. And you're also looking to try to identify those new interactions that way. And then just quantify the host with the images, but not necessarily be able to tell from the image whether or not, and then correlate that to the genetic data to figure out the parasitism.
That's certainly a good point. It's all correlation based, right? So it's some people claim it implies interactions a little more strongly than others. But yeah, I think it's harder to refute because there's so much image data to look through that really, at least so far, I've been kind of just like, where's the diatom? Is there anything that looks weird about this diatom? So I'm more looking for a confirmation if it's there or not. And if I don't see it, I'll move on to something else. But yeah, in theory, this kind of combined approach could be used to kind of validate some of those interactions that people see in these types of analysis.
Thank you.
Great, thank you. Anyone else? Anyone else on online? No. Okay, let's give Dylan a big thank you.